All right, my name is Derek Willis. I'm a second-year fellow, and I'm based at CRED and IRI. So what I'm going to be talking about today is basically there's been a big dramatic increase in funding for anti-malaria programs over the last decade due to the Global Fund and the President's Malaria Initiative. And the vast majority of that funding is going to antimalarial drugs and two different kinds of vector control interventions. One is insecticide-treated nets, and the other is indoor residual spraying. And both of these vector control interventions are pyrethroid-based. Pyrethroid is the kind of insecticide that's being used. And we're seeing growing evidence now that the efficacy of these interventions is going to decrease over time because we see more evidence of changes in vector behavior in response to the intensive and extensive use of these insecticides. And we're also seeing increased levels of insecticide resistance. So while these interventions are very effective initially, their efficacy is going to decrease over time. And at some point, we're going to have to make a policy decision of when to switch over to a new insecticide. So a new insecticide will be very expensive to redeploy hundreds of millions of nets. And it will also involve quite a bit of communication with the public to try to explain why the older nets are not as effective as the newer nets. So what I'm basically talking about today is kind of looking at the current framework we have for collecting information and kind of monitoring this change over time, and propose an alternative framework that's much more dynamic and flexible and hopefully able to keep pace with these changes in vector behavior. So just to focus the discussion on one policymaker, if we take the policymaker in Liberia, if their goal is to suppress malaria cases and malaria deaths, basically they want to use as much as possible evidence from other countries to kind of learn from their experiences, what's worked with implementing interventions, what's worked with a change to different insecticides. So basically we can design a decision framework in terms of which information should be collected from other countries, where the information should be stored, and how to display the information to a policymaker. So in this presentation, I'm just focused on the second two, where the information should be stored, how it should be accessed by a policymaker, and then how the information should be presented to the policymaker, and how should, if we have a simulation to simulate the impact of alternative programs going forward, how should that information be displayed to the policymaker? So just to kind of summarize the current state of insecticide resistance in Africa, basically the red and yellow dots in this diagram are showing where there's already been documented studies showing where resistance is happening or where it's likely to happen in the next couple of years. So basically, we don't really have many studies. We have relatively low number of studies on this problem given how important it is, but there's basically growing evidence that these mosquitoes are not only becoming resistant to the insecticide, but they're also trying to shift their behavior. So the reason that ITNs have been effective in the past is that mosquitoes are biting primarily during the middle of the night, and they're also biting indoors. So we've seen already evidence that mosquitoes are trying to shift their behavior from endophagic or indoor feeding towards exophagic outdoor feeding. Also shifts in their biting time towards the evening rather than the middle of the night. As far as indoor residual spraying, so that's basically spraying the indoors of homes, so after a mosquito takes a blood meal, it rests on vertical surface, then absorbs the insecticide and dies if the insecticide is effective. So in the past, that intervention was effective primarily because mosquitoes were endophilic, so after they took a blood meal indoors, they're then resting indoors. And we've also seen shifts that they're now resting outdoors. So if the mosquito takes a blood meal inside, bites a human inside, and then goes outside to rest on a plant or a tree, then obviously they're not going to be affected by the indoor residual spraying intervention. So basically right now, whenever those studies that had mapped on the previous slide, that came from a Nature paper that came out this past summer, those studies, whenever these come out, they either appear in academic journals or the Global Fund or the WHO. So the Global Fund, WHO have their own set of kind of reporting requirements whenever a country receives funding from the Global Fund. And obviously there's academic journals that are available as well. So the problem for the policymaker, even though there's a lot of literature out there, there's a lot of published studies, a lot of reports, it's very difficult for a policymaker to identify which reports are actually relevant for their problem, for their policy decisions. So if you go to the Global Fund website, they have hundreds, thousands of documents up there. It's very confusing to navigate through that site and actually find the few reports that might be relevant for a policymaker, say in Liberia. As far as accessing academic journals, it's easier now to access the journals without having, without actually paying any kind of fee to have membership with that society. But there's still the same problem of the policymaker having to look through 3 or 4 different journals and finding papers that are actually relevant for the decisions they face. So what I'm proposing as an alternative is basically the development of a web-based decision support system. So this would basically be a website which would centralize this information as much as possible, be obviously free for the policymaker to access it, be free for anyone to access it, and the decision support system itself would actually make it easy for the policymaker to find which information is relevant for their policy decisions. So I feel like one advantage of this approach is basically be much more dynamic. So instead of having to go through the long, tedious process of a report being generated by the WHO or collecting data, then having it peer-reviewed and maybe submitted multiple times, which could take 1 to 2 years, basically this would be a much faster way for this data to be made available and for policymakers to kind of know what's going on in not only their country, but neighboring countries and learn from each other's experience. The website would basically allow policymakers, researchers, anyone in the general public to kind of upload either data, a published article, or even just kind of an anecdotal personal experience with in a research program or their experience with an intervention. So if they have questions about how an intervention is being implemented or ideas about how insecticide-treated nets could be distributed more effectively, that could all be integrated into the website, and kind of like a Wikipedia-like approach of integrating this knowledge and making it more accessible. And one feature we also want to add is basically the cell phone, so ways for people to access the information from their cell phone through text, either through text messaging or through, if they have a data connection, actually accessing a modified version of the site. And finally, what the feature that we're going to have to actually enable a policymaker to identify the literature that's most relevant for their community is basically using the Stability Transmission Index as a search feature. So this is a paper that came out in 2002 by Kashesky and others. And basically this index, the darker the color basically represents the intensity of malaria, the potential intensity of malaria transmission due to temperature, precipitation, and the human biting rate of the vector. So basically, darker regions represent areas where transmission is more intense due to these exogenous risk factors, and the green areas represent areas where transmission is less intense. So if you are a policymaker in Liberia, and you're looking for literature that's maybe relevant to inform your program, you want to look at studies that were conducted in regions with a similar index. So the next question is basically, if we have this information, and we can basically use that information to have a transmission model integrated into the website, and we can do simulations, and we can generate scenarios for the possible impact of alternative interventions, we need to figure out the best way to display that information to a policymaker. So the basis for this study, for this idea, basically comes from a paper in 1981 by Tversky and Kahneman. And what they basically had, they had a survey conducted with students at a university. They said that there's a disease that's going to kill 600 people. You have to choose between disease control program A, which will save 200 people, or B, where there's 1/3 probability that 600 people will be saved, and a 2/3 probability that no one's saved. So obviously the expected outcome is the same in both cases. But in terms of the response they got from students, and this has been replicated many times, 72% of the students chose program A, and only 28% chose program B. So most people felt were more prone to choose choose the safe option, program A, be a definite outcome. So if they reframe that question and had the same outcomes, just framing it in terms of the number of people who will die instead of the number of lives that were saved, basically the decision by these students switched. So instead of choosing the safe intervention or the known outcome, program A, people switched and chose program B. So you can see again, when it's framed in terms of number of lives saved, 72% want A, and in terms of number of people who will die, it's 78% choosing B. So basically, I conducted a survey with Talia Marone Schatz, and basically we conducted a survey with scientists, researchers, policymakers who were members of the International Society of Infectious Diseases. We basically gave them a very similar scenario using slightly different numbers, but basically the same probabilities. We received 1,500 responses to that survey, just trying to see if scientists and researchers respond differently to this question than undergraduate students. And basically in the survey that we conducted, we said that the disease is going to kill 15,000 people. Again, program A, 5,000 be saved, and then program B has the probabilities where you can either save everyone or save no one. So basically very similar, 74% chose program A. And then when we reframe it, presenting it in a loss frame, basically again it switches to 33% and 67%. So the idea here is that basically, even if we have a perfect way of simulating the impact of two different health interventions, there's no neutral way to present the impacts of those, the results of those interventions. And so if there's going to be increasing internet access in Africa, I think for many areas of development, we're going to have to shift towards more of a web-based approach where we can integrate knowledge and make that, and kind of empower policymakers in these countries to make these decisions. But we need to start conducting research to understand how we're going to actually display this information to a policymaker through this website. So a survey that we're planning on doing going forward is basically kind of conducting a similar survey, hopefully once we actually have this website up and running. We want to use the website actually as a research tool to give different scenarios to the users of the website. And one survey that we're considering doing is basically giving them different scenarios for what's going to happen going forward with insecticide resistance. So one option would be just to continue using the current pyrethroid-based insecticides in ITNs and IRS. So the advantage of doing that over the next 3, 4, 5 years is that it's cheaper to do. The disadvantage is that the more we keep using that same insecticide-based intervention, the more resistance is going to grow and grow, and that insecticide may be completely useless down the road. So control B, if we switch to a new insecticide, the earlier we do that, the longer the life of that insecticide, the pyrethroid insecticide, but it'll be very expensive in the short term to actually kind of redeploy all these nets and actually go through all the communication, education aspects of trying to teach people why we need to switch to this new net, even though they look exactly the same. So basically, we want to kind of have a game that people would play on the website where they would kind of go through these different scenarios and try to see if we frame these two different alternatives in different ways, if that really has a big impact on what their preference is between staying with the current insecticide or switching to a new insecticide. So the web-based decision support system, we're already starting to work on it through an Earth Clinic grant. Again, we're planning on using it as a research tool itself in the short term to kind of examine the impact of these framing effects, and then ultimately, of course, the goal is to make this a tool for policymakers in Africa. Just want to mention 2 other projects that we're working on. One is primarily through IRI. They've contracted, President's Malaria Initiative has contracted IRI to conduct an evaluation project of their program in Ethiopia and Tanzania. We're going to submit a report to them in February, and if that goes well, we'll do the similar reporting for other 13 countries in Africa where PMI is operating. And just in the next few weeks, we'll be submitting a proposal to NIH for a study to look at spatial uncertainty. So if we have spatial uncertainty with all these kind of models and simulations, what's the most appropriate visual format to kind of display that uncertainty to policymakers so they can use it to make appropriate decisions? All right, thanks.
Good evening, everyone. Thank you for sticking around. And I'm a first-year fellow here, and my name is Meng Xu. I'm also a postdoc at the Rockefeller University, working with Professor Joel Cohen in his population lab. So today, I'm gonna give you a brief introduction on the Taylor's law, and hopefully by the end of the talk, you can have some understanding of this topic from basically the biological statistics. Okay, and a picture from Taylor's paper in 1961 is here, and on this picture you can see a group of points, actually different groups of points, and lines that going through them. What I want to mention here is each group of points represents the mean and the variance of the number of individual of species, one specific species. So different lines would correspond to different species here. And Taylor actually found out, as you can see in the picture, each group of points lies on the line pretty well, right? And that is what Taylor found out for 24 different species in this case. So this is a graphical interpretation of Taylor's law, and they have a positive linear relationship. So mathematically, I have written down at the bottom here, you can see the mean and the variance and how they are related. So what I just shown you in the first slide is a log-log scale plot, where everything is mapped on log mean, log variance, so it gives you a linear relationship. But in fact, if you want to look at really the mean and the variance, it gives you the power law relationship, right? That's just a mathematical transformation. And some terminologies, you can choose the one you're familiar with, either from the mean and the variance category, okay? Basically, it's the average and the variation. What's the relation between them? So in population biology, we are interested in the number of individuals of a species. At different times, different locations, we can calculate the mean at different times and different locations. Right? So let me give you one example of how we calculated the mean and the variance in this case. This is actually the real data we collected from Black Rock Forest in 2007. And you can see a plot which is 75 by 75 square meters. And there are 9 equal-sized subplots in this plot. And the number in each of them represents the number of oak trees. This is data at the north slope of Black Rock Forest. So mathematically, we could do with what can we do with these numbers? Well, we could take the average, right? That's clear. Biologically, it means the average number of oak trees per subplot, right? So it's give you a density of the oak trees in this plot. And also, we can measure the variation among the number of oak trees in this plot. Mathematically, we can take the variance of these numbers. We are fortunate to have 12 different plots like this, of course, with different data. So you can see the result. We have just finished this last week. So you have 12 data points that each one from one plot. And each plot gives you a pair of mean and variance. And they fit pretty well on a straight line on a log-log scale. Okay. So I have listed the linear regression equation we used here, and you can see the exponent, if you remember in the equation I showed you in Taylor's law. The number 2.36 is the power, exponent in the Taylor's law. So that's the number that we're interested in. Okay, so 2.36. That also falls in the category of the prediction by Taylor, which he predicted to be between 1 and 2. So 2 is not significantly different to this one. We have some test to check that. So from this one, the next 3 slides, I'm going to show you the Taylor's law in different subjects. And this one shows you in probability theory, we choose the negative binomial distribution here. So this picture has nothing to do with data. It has no sample in this one. We just use the mean and the variance, as you can see on the right top corner there, of the negative binomial distribution. And we plot on a log-log scale for varying p and fixed r, the 2 parameters. And they are represented by the blue stars here, and they fall pretty well on a straight line with slope 1.04. So this is one probability distribution that satisfies Taylor's law. And we do have other distributions that do not obey Taylor's law. But just to show you one positive example. This is one from the literature of counting the number of cells. Also, the mean and the variance of the number of cells could be calculated. And this is over 2,000 calculation. Okay, so 2,000 points, mean and the variance are listed here of 178 different species. Here the exponent b is 2.03. Okay. And the last one here is again, Taylor's law representation graphically, we calculate the mean and the variance of the traffic in network routers. Okay, and this one is a different version of the Taylor's law because the mean and the variance in this case are calculated over different times. Right, so we have two versions of Taylor's law. One is over time, which is this picture, and the one we have now the spatial Taylor's law, which is given by the oak tree data, okay, at the fixed time. So you can see the time window are fixed in all these four pictures, 5 minutes. And the time duration for carrying out this experiment are different, runs from 2 days to 2 months. That's why you have 4 pictures for 4 different duration of times. And in this case, the power exponent B is given in the picture alpha. So it ranges from 0.86 to 0.71 and becomes saturated and stable to 0.7 as the time duration become longer. Okay. So just some pictures showing you where Taylor's law holds. Actually, I haven't shown you already both the temporal and the spatial version of Taylor's law. We are able to use this matrix structure to calculate the Taylor's law, or to test the Taylor's law. The temporal one would correspond to, for example, the first row where you have S1 is a specific location, and time is T1 and T2. You take the mean and the average, mean and the variance over different time. You get the mean and the variance, one pair, right? And that gives you the temporal Taylor's law. If it satisfies, we don't know, right? I've shown you some data they satisfy, but that's how we test it. For the spatial Taylor's law, at the fixed time, you look at the first column, A11, A21 are the numbers of individual species at different locations, S1 and S2. You could get the mean and the variance over these 2 locations as well. So 2 versions of Taylor's law. Using this matrix structure, A11, A12, A21, A22, just a 2x2 example, but here, again, from probability theory, we used Poisson distribution with the same parameter lambda is equal to 1, and then we were able to show the Taylor's law holds simultaneously both spatial on the spatial scale and the temporal scale, but they do have different power exponent. Spatial one is with the slope, as you can see, it's almost 2 there, and temporal Taylor's law is about 1. So this is just a mathematical finding of the temporal and spatial Taylor's law. Later we want to see what the real data tell us between these 2 versions. So my last slides, I have shown you a couple of examples where Taylor's law holds, right? The question in the middle. Whether it holds, in the population biology, we still don't know that answer to that question. But so far, from the data we collected in the Black Rock Forest, they do give us a very good fit for the Taylor's law. And on the top, the biological interpretation of the exponent, we are particularly interested in B. We still don't have a quite good biological understanding of the exponent there, right? And more importantly, I want to talk a little bit more on the practical application of this Taylor's law in pest control. You want to measure or estimate the pest density in certain areas at a certain time. And the way to do it, you have to design your sampling so that you can save time and expense to measure the real density data. And that involves Taylor's law, if Taylor's law holds. So you can use Taylor's law to develop the algorithm to help you design the sampling so that you know where is the most dense pest population. So you can do certain timed actions, like releasing enemy species, or you can spray pesticides over that certain area. The last thing I want to touch on is the links between the population variability is given by the variance mathematically and the individual and the ecosystems. So far, we have also looked at the data from Black Rock Forest on the individual level. We look at the oak trees, and we found actually verified the allometric relationship between the average body mass of the oak tree and the oak density in that area. So that is individual level allometric relationship. And if the Taylor's law holds, as I have shown you, for the variance of the number of oak trees and the density of the oak trees, then we can connect these two to directly look at the population, the oak tree variability, the variance, and the individual average body mass. So that's the message, and that's the future work for us to do. And that's all for my talk. Thank you very much.
Thank you. So we'll make time for some questions. And Jeff, would you like to have the first questions? Since I know you have.
Yeah. Great. Thank you, and apologies for the first group of presenters because I wasn't able to get here in time. And apologies for having to leave early because I have to go catch a plane to Hong Kong, unfortunately, for some of our Millennium Village work, which will be featured by Ericsson in a global conference that I'm going to. And absolutely wonderful presentation, so I want to thank everybody. I had a few very brief questions that I wanted to ask for each of the presenters. First for Daniel, the question about the commercial thresholds for this to be a viable economic system. Two questions. One is, what have you estimated yet what you regard as the capital cost threshold to make this system actually reach commercial viability in terms of pricing? And second, if you stick in a diesel generator rather than the solar, what are the comparisons of that, still with the wiring and the prepay and so forth, but with a different power source for the local system? For James, I've always been told, you're very fascinating talk, I had always been told, maybe complete myth, that the seasonality of influenza was that in winter people are more inside, and so the beta coefficient in the SIR model of number of direct contacts per index case goes up. And you're giving a biological explanation, and I just would like you to comment on the different interpretations and what is the prevailing idea or ideas, or is it complete mystery, this question, but this was the first I heard of the biological explanation. It doesn't seem to work. Well, I mean, it could be biological with this U-shaped relationship, but that poses a biological hypothesis, which you didn't discuss. And could there be a more sociological explanation in the tropical zones that the epidemics seem to occur, or the seasonality is during the wet season? And could that also have some explanation in terms of face-to-face contacts and transmission, interpersonal transmission dynamics? For Catherine, actually, could you just say a little bit about the environmental burden of the nitrogen versus the phosphorus leaching, and in terms of eutrophication or other kinds of effects? Second question was, you talked about planting trees as leading to less leaching, but maybe we're interested in, and I can imagine just more biomass of different kinds drawing off the nitrogen, but we're mostly interested in somehow the amount of leaching per crop achieved. So there's a kind of productivity measurement. If the system is productivity is reduced by having additional biomass, it may be a thin victory. If it's increased, it is then could be noted. And my last question, just technical question, is leaching the only way that the nitrogen deposition occurs, or can it be through volatilization of the nitrogen? When we think about the dead zones, is it all in situ leaching of the fertilizer use, or are there other ways that the nitrogen cycling collects the nitrogen in the estuaries other than the direct in situ? Because there is also physical movement of nitrogen through the air, I guess, as another part of the nitrogen budget and the nitrogen process. So those are just questions that I had. For Derek, where's Derek? Yeah, hi Derek. First, a suggestion. I don't know if you're in touch with Prabjot Singh et al., who is working on, also in the Millennium Village context, on data collection, for ways to collect dynamically data on, for example, incidents of infectious disease, and it could be malaria, as indicators, real-time indicators of epidemics. There's a large project on trying to aggregate different kinds of internet-based information or tweets or other things to try to identify relevant public health information. It just seems like there should be something there. And I'm always asking the IRI malaria team to work with the Millennium Village malaria team more closely to get that integration going. But it did seem to me the phrasing of the questions, I think, is very interesting. Of course, the prospect theory and the loss bias. But also, when we talk about information for policymakers, it wasn't clear what kinds of information you want them to be, you're thinking they should be looking at, and not only how they should be evaluating it, but which information in real time would you like them to be seeing, and what can they do about it? So if you tell policymakers that there's increasing pyrethroid resistance, that by itself doesn't indicate any action options for policy. And so it's not really a policy relevant fact until it has a policy relevant interpretation. And so I think for policymakers, they want to know what the information means in terms of options, choices, how to evaluate, not only telling them pyrethroids are becoming more resistant or mosquitoes are becoming more exophagic, but if at all, whether there are things that they ought to be doing about it. And often it's not obvious what they should be doing. Maybe the question is, when do they switch over to a new insecticide or something else? But I'd like more clarity from you about how the information, not just how to evaluate the information, but how to identify the action agenda that is implied by the information. And for Mengshu, thank you very much, for very interesting. You didn't really say much about how, I think it was the 2 points along the curve. One is the probability model. You showed how a negative binomial or a Poisson might generate a Taylor's law, but you didn't really say much about how that might be relevant as a biological interpretation. So presumably it could be something about seed distribution or successive fertilization of seeds or something else that would have one of those probability models attached to it and therefore lead to a Taylor's law. But I wondered if you'd say, give an example where there is a biological interpretation that comes from the probability distribution underlying this. So I'm going to wait for those answers. I'm going to have to go. So let me first thank all the fellows. And again, I'll look through the presentations once they're online of the ones that I missed. And I do apologize that I came late. This is our really most critical program, the postdocs, because you are the source of the scientific progress. And this program has generated some very big things also for the Earth Institute. We have a major Gates Foundation program on global monitoring of agricultural landscapes that really emerged from the fellows program, because we had 3 fellows who said we need to be monitoring agricultural landscapes more closely, and they created an international conference which was hosted, and that led to a Nature article, and that led to the Gates Foundation being interested, and they hosted something, and then with Cheryl, who's now leading this with the team, that has led to the beginnings of a major program. And Cheryl, it's fair to say it started from this group. This was completely Lee Winowicki, Sean Smuckler, and Rosalind Riemans, right? We're the 3. And so I just want to encourage you guys, take your big ideas and come to us. We can host events, we can organize meetings, we can organize workshops. The resources are there for you to work together, brainstorm, promote from your work or from the cross-fertilization of your work, further activities. And multi-million-dollar global research programs can emerge from that. So don't be hesitant to think big. And one more point that I just want to put into the fellows' thinking, because I want to come back to you about this for some brainstorming. We're now about just 3 to 4 years away from the end of the Millennium Development Goals period, which was 15 years from 2000 to 2015 to make major reductions of poverty, disease, and hunger. And there's been a lot of progress, but not enough to achieve the Millennium Development Goals in a lot of places, though we think that much faster progress could be made if the investments were targeted and intensified. There will clearly be a post-2015 global agenda, and there's talk now of creating, instead of Millennium Development Goals, Sustainable Development Goals. So one idea is a move from MDGs to SDGs, and we as an institute will definitely be involved one way or another in the discussion of defining the post-2015 Sustainable Development Goals and thinking about programmatically the major research questions and the major avenues to achieve them. And a lot of you are working on critical pieces of what post-2015 sustainability would mean. For example, the urban environment is clearly one of those. Sustainable agriculture is obviously at the center. Sustainable energy systems are key. So one can think about several different dimensions that might become the next round of goals. So I think that this would be fun for the fellows to get engaged in, and we'll organize a meeting together, and I'll describe to you some of the international diplomatic processes going on, and we can brainstorm a bit about that. So I'm going to sit down, and I'd love to hear the answers to just those questions, and then I know there'll be more discussion, but I'll slink out after that so I can catch my plane. But thanks a lot.
So maybe you can go in order.
Okay. So the question, as I remember it, was at what sort of cost point could these sort of solutions scale? And then what are the differences between in costs of having a diesel generator and then some sort of renewable power system? The first question I can't answer directly. It's something we're working on. What I can tell you is that it's dependent on a couple of things. It's pretty sensitive to things like the density of the households together, because the wiring costs can be immense. And it's also highly dependent on, at least for solar, the solar resource. And then the third dimension is really the purchasing power of the people, so that, because that is going to mean a larger revenue stream for you to amortize the initial investments over. So given that, what we're focusing on for places where this might scale initially is peri-urban regions in sun-drenched countries, basically. And then actually being in the Millennium Villages and the places are really the most difficult to reach, but the data that we're getting is going to be pretty interesting. On the piece about the diesel generator versus solar, solar is definitely more expensive from a capital expenditure perspective, but there's also going to be, you still have to wire the homes, and all of that labor and material is actually a pretty significant fraction of it. So in some ways, if you just look at how incredibly cheap a diesel generator is, you might get the wrong idea. But when you really look at the holistic cost, the incremental sort of cost of having these renewable systems is not a minute fraction, but a smaller fraction than you might expect of the total budget by the time you've actually wired everybody, put in light bulbs and sockets.
But I guess it would be really interesting to do a full, full analysis and the levelized costs. So assume a manageable interest rate of 6 or 8% on the capital expenditure side. Take into account the differential renewable, the differential costs of recurrent costs of the diesel, plus transport and other unreliability and so on, and really make an evaluation of in what places would you have the same system but a diesel generator there and the monitoring versus the solar array.
Stay tuned. I was chipping away on that spreadsheet this afternoon.
Good. Great.
Daniel, how much was the total installation cost for Uganda and Mali Millennium Village? And how much was the revenue collected over the past year?
Okay, so the first one is unacceptably high, but that's because it was all off-the-shelf research systems. The metering electronics for us was on the order of $5,000. The solar generation was on the order of $10,000. Again, if you did this at scale, you should be able to do a lot of these things cheaper, and a lot of the things we did, you wouldn't want to follow the same approach. A lot of the decisions we made were in the name of expediency rather than scalability. And the second part?
And the revenue you collected from the village people?
People are spending on the order of a few dollars a month each. Each household, not people.
So you're covering 200 households, right?
Can I ask that the fellows go to the front so that we can use this mic for audience members? So that the recording actually gets your answers. Thank you.
All right, Jeff. So you were talking about a little bit about influenza seasonality and the thought that you had heard that indoor crowding during the winter was the cause of influenza seasonality. And that indeed is very pervasive in the literature, going back until about 10, 15 years ago when people started looking at this a little bit more. And I would say that it's not necessarily a myth, but an assumption people made and never really studied. And it doesn't quite follow once you start looking at the data. Once you start looking at how people interact with each other, we are on public transportation in crowded places such as today all the time, during the summer, during the winter. And also, if you look at warmer climates, such as I did my doctorate in Tucson, Arizona, where during the summer we are indoors all day. In the winter, we're outside enjoying the nice temperatures and stuff. In the
Same cycle.
Yeah, exactly, the same exact cycle. So there's something probably else going on. That said, school cycles have been shown to have a big effect on transmission. So when kids are in school, there is a higher level of transmission. But there seems to be, the evidence suggests that there's some interaction between that and some climate or environmental factor. And so in this case, I was showing the relationship between absolute humidity, virus survival, and transmission. But others would argue that there's other hypotheses out there, such as vitamin D, solar radiation, effect that has on vitamin D and immunity, and yeah, many others. So the question about the U shape now, so it doesn't follow in the tropics that we have, okay, that the epidemics are occurring in the most humid time of the year. There are a couple influenza virus survival studies that actually do show a U-shaped relationship between humidity and influenza virus survival, although the majority of the studies show a monotonic relationship. So that doesn't quite explain that. So the other possibility, which I think you might have been alluding to, was that there's some sort of confounding going on, that it's the humid season, which is also the rainy season there, which is true. So there's actually, in the tropics, influenza occurs during the rainy season. So there's really no mechanism explaining that right now. It might be crowding, but again, that conflicts with the temperate place patterns. So yeah, so it's still a question.
Good problem to crack.
Yeah, yeah.
But, all right.
We go to Kate.
So the first question was just in general eutrophication and how nitrogen and phosphorus. So nitrogen and phosphorus are limiting in different systems. So in temperate systems, we think of as being nitrogen limited, and phosphorus as being limited in tropical systems. So it kind of depends where you are in the world, right? So if you're in a nitrogen limited system and you have a lot of nitrogen leaching off, you're going to see eutrophication in those regions. So and in that picture of the dead zone, it's a combination of both, because you're starting to get a little bit more southern, so you're starting to also have phosphorus limitation as well. So a lot of it will just depend. And then of course, if you're in a nitrogen limited system, what are you going to apply but very strong nitrogen fertilizers and vice versa. Although, granted, in the tropics, any time that you are planting a crop, you have to put on a lot of nitrogen, because everything is in this stoichiometric balance. So you have to make sure that you have enough of everything to keep productivity going along. And then the second question was about there were three. Yeah. Oh, I remember. So if you plant too many trees, you're going to shade out your crop, right? So one of the cool things about the figure that I showed you, that sort of exponential decline, was that at some point you can imagine, like, you get this point of diminishing returns where you plant enough trees, okay, good, and then you can imagine sort of another curve going in. So there have been studies, of course, on what happens when you increase the amount of shade cover. And so, you could imagine putting these two together and trying to find some sort of optimal amount where you're both benefiting the system because you have the trees that are sucking up all of the nutrients, and then also you're not overshading. And then I should say that these trees are remarkably effective, and so we may find that you don't even need to plant that many, or as long as you have, a lot of the trees on farms are not necessarily intercropped, but they're around the farm, which can obviously, if you have a slope, they provide this barrier, this buffer zone. So that can also be important. And then the third question I definitely need to...
Sorry. Oh, yeah, just about the nitrogen budget, whether volatilization other than leaching.
Right. Okay, so volatilization is not necessarily my area of expertise. I work a lot with water. There is a graduate student, graduate student, I'm still in that mode, a postdoc who is actually an EI fellow, Jonathan Hickman, and he does a lot with volatilization. And sure, that can be a major player, but when we're talking actually about eutrophication, usually we're talking about what's the groundwater, because nitrate is, it's super mobile. It latches onto stuff, it moves through the system really quickly, and that's the other difference between nitrates and phosphates. Of course, phosphates, they form really strong bonds with mineral soils, with clays, and so, because they're -3 and nitrate is -1, so they just have different different patterns. And when we talk about eutrophication, mainly we're talking about what's happening with water. Okay.
Okay, so thanks. So just the first comment about working with the Millennium Villages, we're actually going to use the data from Tanzania and Ethiopia from the villages in the PMI evaluation. So I've been meeting with Paul Pronick, so that looking forward to using that data. Actually, Rebecca Smith is sitting there, is going to do a lot of that analysis. As far as, I think it's, in terms of which information to provide to policymakers, I think it will be very much determined by which policy decisions they're allowed to make. And I think even though the Global Fund says that these proposals are coming from these country coordinating mechanisms, there's a survey done in Tanzania where they actually asked policymakers what has the most influence on your proposals, and they said actually the donor does. So they actually basically know the donor's only going to fund this, so we should submit a proposal for that. And I've heard that from other policymakers as well. So I think that needs to be addressed, because I think right now the national malaria programs in a lot of these countries are basically just being told what to do, and they get the money and they implement, which is better than no money, but I think there needs to be more kind of independence for countries to be making these decisions about when they want to transfer to a new insecticide, when they want to do different interventions. I think the WHO is planning on a big meeting, I guess very soon, to come up with like kind of a global strategy for switching to new insecticides. So I think that's great, but if it's a kind of a global effort to do that, I think it's going to take a long time, and if they're going to say everyone does it at the same time, I guess that's what will happen. I think hopefully if this information is available, if these policymakers are more empowered with this information, maybe there'll be more flexibility from the Global Fund to give them more leeway with those decisions. I think definitely the IRI data as far as temperature precipitation forecasts is really definitely something to integrate into the website, especially in areas of seasonal transmission, to kind of forecast when rainfall will be happening. I think no matter what, I think there's a lot to be gained from just policymakers learning from each other how to implement nets or spraying using fewer resources. I think there's a lot of ways to become more efficient in that process. We shouldn't think in a very static way about these costs. A net $6 everywhere. It should vary and should decrease over time if we learn how to do it better. And as far as other interventions, the Global Fund's not doing this now, but I think especially in areas in Africa where there's a lot of urbanization happening, we need to think more about source reduction interventions as being a possibility. So either larviciding or environmental management. It's a lot cheaper to do that when the city's in the initial stages of urbanizing than trying to go in when it's 3 million people and there's huge people, 100,000 people living in a slum. It's very difficult at that point to actually try to move people out of that area if it's a high-risk area with a lot of standing water, or trying to put in drains. So I think if we kind of think, again, not think about environmental management and source reduction being a very static cost, recognizing if we do it early, it'll be cheaper, and also if we're learning from each other's experiences, I think that cost can actually decrease a good bit.
So let me repeat your question so I want to get it right. You asked about the probability distribution that I show that satisfies Taylor's law, and you want to know whether there is biological meaning, right, behind the mean and the variance of that. So to that question, I think the short answer is no. I haven't done that, but I think the starting point would be the exponent b there. As I'm showing you, is range from 1 to 2 roughly, and we did have seen some probability distribution that give the same range of exponents, and I have shown you a couple of experiments that give you the same type of number, right? So if they give the same number, whether can we use that distribution to interpret the data set we have? So that's one starting point, and I think that would direct us to the modeling part to build up more complicated models, not just the probability distribution, and stochastic differential equation maybe. And but that's for future work. We definitely want to take a look at. Yeah. Thank you.
So I think what I'm going to do is not have any more questions, because we have a little eating and drinking thing to happen now. And I know you can ask questions while you're eating and drinking. So if people have remaining questions, first, thank you, Jeff, for making the time to come amid your busy schedule and asking such wonderful questions. And thank the postdocs for making the presentations. We do this every 6 months or so. So about 6 months from now, you'll see a slightly different group. Some of the same faces, some new faces, the same sort of format, probably not here since we change every time. But you're very welcome to come. If any of you have questions about applying to the postdoc program, I won't go through the 9 yards of that, but if you do have interest in that, let me know, and I could advise you on that. This is the best postdoc program in the country, so the best people in the country should apply for it. And we have the best people in the country. Thank you all very much. Thank you.